So, our discussion on neuroglia. So, we now discuss about the astrocytes. We discuss the astrocyte in a very detail. In recent year, means last 20-30 years, it's astrocyte only neuroglia which is study very well, but it is also under study. But whenever there is a history of neuroglia, it is a find out in 1856. So after many years, means 100 and 150 years, 150 years, till now no one are interested into research of neuroglia cells. But now recently they show some interest in neuroglia cells, especially astrocytes, because a scientist thought astrocytes are not only, or means especially astrocytes, neuroglia cells, they also give us some functions like learning. They are also responsible for some conductions. Also they are responsible from brain activity. So they now they are under process of study of astrocytes. So they, whatever study we have astrocytes till now, we will discuss, but this is not given in a book. This is a not given in book. I refer from international journals, from different international journals, or scientific evidence. So astrocytes, basically it's a star-shaped neuroglia cells present in a central nervous systems. We know there is a two types of astrocytes, fibrous and protoplasmic. Fibrous are remain in a white matter, protoplasmic remain in a gray matter. Fibrous, it's having a long process or very less branching, but protoplasmic having a very much branching, but it's a short branch. This is all we know about the morphology. Now astrocytes, first we talk about the some functions, very basic functions. Astrocytes having a branching. I draw the one astrocytes here. This is the astrocytes. It's a branching. So the food process, or this is a branching, or it's called as a food process. Some food process are attached with the capillary. Suppose this is a capillary. Some food process attached with the neuron. Suppose this is a neuron. Some food process attached with the pyometers of meningeal or covering of brain. Means there is a, I draw a pyometer like this. This is a covering. So basically they attach with the capillary, like I draw the foot. They are also attached with the neuron and foot with pyometer. So with, they give a first functions, they give a support, because they attach with the multiple parts, multiple areas. So they first is a physical support. Second functions, that they no, also they foot attach with the blood capillary, means they are responsible for formations of blood brain barrier. That is a BBB. They are responsible for formations of blood brain barrier. Though they nowadays study shows this food pressures of astrocytes, they are not a true barrier. Okay, but they produce some substance which are responsible for formations of tight junctions between two endothelial cells, and which is actually a BBB. So they are responsible for blood brain barrier. Next, during intrauterine life or during development, they produce some chemical substance which are responsible for neuron growth. So they are responsible, they secretes the some chemical factors, it's called as a neuron growth factor. Different type of neuron growth factor which are responsible for development of neuron, which is true cells. So if astrocytes are deficient, they cannot produce the sufficient amount of neuron growth factor. So they are responsible for neuron growth factor, and they are responsible for growth of nervous system. Okay, next functions, they are also make a synapse, formations of synapse. They are around the synapse. Now what is synapse? Synapse is a junctions between a two neurons. Suppose this is the axons of one neurons, they end, and from here there is a next neuron dendrite start, and again axon. So this a microscopic structure or microscopic gap between two neurons, this is called as a synapse. So this astrocyte food process, they also remain here. So they also responsible for synaptic function. What type of synaptic functions? They are responsible for reuptake excessive neurotransmitter. Whatever neurotransmitter remain in synapse, if it is excessive, they reuptake by astrocytes. So they are responsible for synapse formations and also responsible for excessive neurotransmitter reuptake. Also, next functions of astrocyte is they are maintain the ionic balance around the neuron, especially level of potassium. Whatever potassiums are excess in around the neurons during depolarizations. So these potassiums are taken by astrocytes. I will explain how in a few minutes. So they are also responsible for ionic balance. Next, astrocytes are responsible for repair of neuron injury. But it's a surprisingly seen in the central nervous systems, whatever neurons are damaged, they are scarred by, or they produce the scarring effect by astrocytes multiplications. Whenever neurons are damaged, astrocytes prevent the neuronal regenerations. It's a surprisingly seen. But while in PNS, whenever neuronal injury occur, they are immediately regenerated by Schwann cells. So this is a controversy that astrocytes prevent the neuron regenerations in a CNS, while Schwann cells promote the neuronal regenerations in a PNS. Why it's happen, we don't know, but basically a theory or hypothesis is that whenever neuronal injury is produced, they produce the apoptosis. Means neurons undergo self-suicidal death. They also produce some proteolytic enzymes. So to prevent this further damage of inner cells, they are prevented by scarring of injury by making astrocytes. So astrocytes can multiply. So in a central nervous system, when astrocytes can multiply without any reason, unnecessarily, without any requirement, they produce a glioma, astrocytoma. This is a very much malignant brain tumor. But normally they are produce scarring whenever there is a damage. Whenever damage in neuron, then and then only they multiply and they produce the repair by scarring. But they cannot promote the neuron regenerations, neuron repair in a central nervous systems. So this is about the repair. Also, they give a glucose, or they give a nutrition to the neurons as a form of lactate or lactic acid. This is the most recent discovery that whenever persons, there is a brain is like a hungry man who always eating something, because they require the more and more glucose, because they active for long time. This astrocyte, because we have a multiple neurons, we undergo, they getting a multiple sensations from all over the body. So they all time active, metabolically active. So they require the more and more glucose. So it's like a hungry man. Brain are always hunger for glucose. So whenever persons doing some extra work or exercise or hard work, that time brain required the more and more glucose. So this glucose cannot transport by a circulatory systems in enough amount. So that time astrocytes produce the glucose, not glucose, but they produce the glucose by glycogen store, which is stored in a astrocytes. So they produce the glycogen breakdown, or they convert the glycogen into glucose, and they deliver to the neuron. But they also deliver as a lactate form. They convert the glucose into lactate, and this lactate are utilized by neuron during exercise. This is seen in a rat. This is a discovered by a Japan neurologist. It is described in a journals of Japan Neuro Association of Neurology. So they give a glucose or lactate. This is a most recent theory that astrocytes store the glycogen or glucose as a glycogen form, and whenever neurons required, they deliver to the neurons. So this is the functions of astrocytes in a short. Now astrocytes can signal with other astrocytes or neurons. Formally they are thought that neuroglia cells cannot give a signal to each other, but recent study shows that astrocytes can be signal. They make communications with other neuroglia like astrocytes and other oligodendrocytes by a signaling molecules like ATP and other like a glutamate. So this basically secretes a signaling mechanisms or chemical. This is called as a glio. There is a neurotransmitter. Neurotransmitter means a chemical which is secreted by neurons to give a message to the next neuron. So this is called as a gliotransmitter. Gliotransmitter means a molecule or chemical or messenger molecules which is secreted by a neuroglia cells, especially astrocytes, and they give a message to the next astrocytes or another neuroglia cells, and they give a signaling mechanism. They give a communications. So basically astrocytes, suppose I draw only the one astrocyte and one food process, and here are the next astrocytes. Okay, zooming this, they are connected by a gap junctions. What is gap junctions? It is a types of synapse, but between, suppose synapse is what is synapse? This is the end of neuron, and this is a starting of dendrites. So there is some space, microscopic space. This is called as a synapse. This is the chemical synapse, types of synapse. This is called as a chemical synapse. But whenever there is a no microscopic gap, this is a 1 and 2 neurons. It's not neuron, it's a neuroglia cells. Between this there is some connections, tubular connections, a microtubule. This is called as a connections, and this is a filled with the water. This is a aqueous microtubule. So whatever substance here, they can easily cross here. This is called as a gap junctions. But there is not truly gap, but they are connected with connections, some microtubule structure. Or this is gap junctions. So microglia or astrocytes are connected with the gap junctions with another another astrocytes. So there is a signaling mechanism is ATP, or also there is a gliotransmitter, we call it a gliotransmitter ATP. Also there is a glutamate, which is also neurotransmitter, glutamate. So how they work? So suppose this is I draw the larger diagram of astrocytes, two astrocytes. This is a one, and here the second, and they are connected with a microtubule. This is connections. This is aqueous tubule. And one more thing is astrocytes are responsible for vasoregulations, vasomodulations. They vasomodulations means they produce the vasodilatations as well as vasoconstrictions in a central nervous system by a producer some special chemical messenger which is secreted by astrocytes. So which is generally seen in a MRI. Blood flow to the CNS are mainly depends on astrocytes. So they are now again talk about this. Whenever some stimulants occur, whenever some stimulants occur from here, they produce a internal messengers IP3. This is called as inositol triphosphate. Inositol triphosphate, which is a chemical messenger between two neuroglia, two astrocytes. What these IP3 do? They stimulate the internal organelles, especially endoplasmic reticulum. You know what is endoplasmic reticulum? It is a tubular structures which is filled with the fluid and massive amount of the calcium. So whenever IP3 are increase in a intracytoplasm or cytoplasmic IP3 increase, they stimulate the organelles. What they do? They bring out the calcium outside from organelles. So what happen? Cytoplasmic calcium level rise. Because what they do, IP3 stimulate the organelles, especially endoplasmic reticulum. So this endoplasmic reticulum excrete the calcium from endoplasmic reticulum to the cytoplasm, means inside the cell. Intracellular calcium level increase. So whenever this intracellular calcium level increase, they also stimulate the more formations of IP3. So in this first number of cells, IP3 level increase, calcium level increase. This IP3 and calciums are now, they can cross, they can travel from these microtubules to the next cells, second number cells. What they do here? Here now IP3 is here, also calciums here. So this IP3 also stimulate the endoplasmic reticulum of these cells, and ultimately they also increase the calcium level, more and more calcium level oozing out from endoplasmic reticulum, and further go on up to the terminal. Means by this way they communicated with one cells to another cells. They are stimulated, they produce the IP3, intracalcium level rise. This IP3 and calcium stimulate the next cells further. So by this way calcium waves occur. Suppose this is a 1, 2, 3, 4, 5 cells. Here the calcium rise, again here the rise, up to the terminal. So by this way they produce the calcium waves, and this is called as a communications. This is like a action potential. This is a study very recently, and up to the terminals, when the calciums rise, what they do is produce effect. A calcium sensitive neurotransmitter or vesicles, they secrete the neurotransmitter or gliotransmitter in the next or last terminal parts, like a glutamate. Glutamate is a calcium sensitive vesicles which is stored in a terminal part of cells. So by this way astrocytes work. So it shows this connections is important. Okay. Now what this glutamate do? Means they are responsible for synapse formations. We talked about the functions of astrocytes. So what is synapse? Synapse is a connection between two neurons. Suppose this is a and here the next neuron, it is axons and it is dendrites. This is called as a presynaptic neuron. This is a postsynaptic neuron. So what astrocytes actually do? Astrocytes they make a wrapping around this synapse, especially in postsynaptic neuron. So whenever this glutamate are secreted here, they stimulate the postsynaptic neuron or also they secrete the more gliotransmitter like a GABA. So by this way they produce some actions. They modulate the action potential. They can modify the action potentials. It is shown in a patients of epilepsy. This connections, this GABA cannot produce by a astrocytes. So this post-synaptic neuron are stimulated more and more. You know, this glutamate is a stimulatory or excitatory neurotransmitter, while GABA is a inhibitory neurotransmitter. So they are responsible for, this is a three parts. This is called as a triparate synapse, means three portions: presynaptic neuron, postsynaptic neuron, and astrocytes. This is a triangle, triped. This is called as a tripartite theory or synapse. They modulate the synaptic connections. They modify the some impulse by this way. So this is shown in a patients of epilepsy, patients of schizophrenia, patients of Alzheimer's disease, that astrocytes play a role in this all conditions. So this study still going on, and after this study our view, our vision toward this disease may be changed, or pharmacological or drugs may be changed after a full discovery or full research. So this is the end of astrocytes. Hope you like this lecture. This is a recent theory. This is not given in our book. So please make a notes from here. Okay. So lecture over.